Thank you very much, Mohammed. Such a great pleasure to be with you today and to be with all of these great scientists, actually, to talk about genome editing and how we as Merck can support you with that. So a brief introduction of myself. So my name is Widad, and I am the application specialist for Africa and Middle East for the biology and diagnostics portfolio from Merck. So I'm based in Tunis, Tunisia. And like I said, very happy to be with you today. My presentation is going to be a little bit more general. So it's not going to be focusing on zebra or zebrafish, but it's more about the genome editing in general. So what we can offer, what we can support you with, and also a little bit back to basics of the evolution of genome editing and how does the workflow look like, and what considerations to take into account in order to have the best result possible. So just actually this slide shows actually the importance of functional genomics overall. So functional genomics uses all the tools, bioinformatic tools, the genetic tools, proteomics, metabolomics, everything. So in order to try to understand this relationship between genotype and phenotype. So, as you know, it's very complicated. That's why we are here today to discuss one of the great models actually that are used for to study functional genomics, which is zebrafish, but to show actually the complexity of trying to understand this very not easy relationship between genotype, phenotype. So, of course, we started, usually we start with DNA and looking for the mutations and inducing actually mutations using the genome editors, mutagens, fusions, insertions, RNA, so looking at the epigenetic modulators, and then looking at the protein, the effect on protein. It's all of this actually to try, like I said, to understand this relationship between phenotype and genotype, and try to find, mainly cures to lots of the diseases that we are seeing, and try to understand lots of phenomena that we are seeing also. So this is a brief actually overview of what we have been doing as Merck for over the last year. So we started with genome editing since a long time now. So with supporting researchers with mutagenic chemicals. So we until 2009, where we introduced the first generation of genome editing tools like the zinc finger nucleases and the talents, till the discovery or the development of the next generation genome editing, which is, CRISPR-IA, the prime editing, the RNA editing. So now gene editing or genome editing, and now we are even talking about the epigenome editing, not only the genome editing. So it's about knocking in, knocking out, but more precisely now. So we are talking about the fine tuning or the fine-tuned modulation. So where you just actually increase or decrease the level of expression of the gene till you get the desired outcome or the best actually dose of the gene, of your gene of interest. So there are multiple tools actually that allows the modulation of the expression. So it can be via RNA, it can be via DNA, but RNA, it's of course, here, as you can see, so the single interfering RNA, so the HHRNA lentiviral infection, the mission, so this is one of the recent products that we have developed, so the mission HHRNA plasmid. But this is, as you know, so when we talk about RNA, so we talk about the next step after the DNA. So we are not really talking about the modulation of the expression at the DNA level, but it's at a later stage via RNA. So, but then lots of things have been done to modulate expression via DNA. And theoretically, when we talk about DNA, theoretically, any change can be done. So anything is possible from the transcription factor. So adding, deleting transcription factor binding sites at the promoter level, so deleting, mutating the promoter at the first exon, at the actually different the next exons, at the three prime untranslated region. So theoretically, everything or any change is possible on the DNA level. And here so comes the the most famous, or the first actually tool somehow that was developed actually, which is the zinc finger nucleases, which are specialized molecular seizes that have high precision but low flexibility. So they target a unique DNA target in the genome, so no related sites, and need an accessible chromatin. So this is to show you actually the CRISPR system. So it's made basically of the track RNA. So, and the CRISPR RNA, which is a sequence that is complementary to the sequence that you want to modify or to change. So both of them together, they make the guided RNA. And of course, they need, so the whole, the enzyme using the guided RNA, so they need all of them to make this complex in order to perform the change that is needed actually, and that you are looking for. So yeah, 10 years ago, so the CRISPR gene editing was introduced to the world by the first paper that was published in Science, and then 8 years after, so all that, Jennifer Doudna and Emmanuel Charpentier, so they shared the Nobel Prize for Chemistry. So for gene editing after that. So this shows that the importance actually of gene editing or genome editing in the drug development in lots of things, and improving actually now, as you know, so we are going through a lot of global changes when it comes to climate. So we are even talking about, of course, we're talking about plants that are resistant actually to the climate change. We are talking about also animals that are resistant to climate change. So, and we are talking about, all kind of things that needs to be, of course, within the what ethics allows. So anything that can be adapted in order to help us survive in the next coming years and centuries. So CRISPR is more than, of course, endonucleases are more than molecular Caesars. So this is an illustration of what what is it actually to do a knockdown or knocked out, and inactivating, inhibiting actually a gene expression using CRISPR. So the workflow. So for anyone who is now thinking of starting using gene editing, so of course the workflow is made of, I would call it, two main steps. So the first step includes several steps, so which is designing the experiment, and then evaluating and validating the experiment, creating the model, assessing the model. So this will go like in a circle. So you have to, of course, assess the performance of your model, and then if it's not doing well, so you have to redesign the experiment, reevaluate, create, and assess before moving into the translation phase. So designing the experiment. So here, of course, you have to select what you are looking for. Are you looking at large scale? Are you looking at single cells? So, and here, so the more, the more you decrease your scale, you more increase the specificity. And here, of course, so we can support you with, if you are looking at the whole genome, a few gene pathways, specific gene families, individual genes, and even the single cells level. So, okay, I'm looking at the time. So here, so we can support, by two, if you are looking at choosing, if you are looking at, multiple hits or one hit. So here, so we can support you with pooled library screening, so where you can have access to 1,000 guided RNAs in one tube. So, and for this part, or for this way, so there is I mean, of course, you will need an antivirus. So whole genome can be screened efficiently. There is a possibility of in vivo screening. But at this stage, of course, the convolution, so NGS is required to analyze data and to identify it. So this is, but this is unlimited options, have limited options for phenotype and readout, cell death or proliferation, sorry. So, but there is also a possibility of the arrayed library screening. So here, so you have one guided RNA per well, multiple format options available. But the time to screen increases with the number of clones that you will be selecting. The in vivo screening is not possible, in contrary to the pooled library screening. No NGS required to understand the results, and multiple options available for phenotype readout, for example, fluorescence, luminescence, high content imaging. So yeah, if you remember 2 slides ago, so we said that you can start from the whole genome down to one single cell. And here, if we are talking about the single cell level, so we are happy to introduce this, the novel optimizable barcoded sgRNA effectors that we have developed using in partnership with 10x Genomics, so that will actually allow you to get into the single cell level analysis. But of course, I mean, so either you are looking at the complete knockout, the decrease in expression, or increasing the expression. So we are happy to let you know that we can support you with all of that. So if you're looking at the complete knockout, so we have the, Sanger arrayed CRISPR KO libraries. We have poison exon knockout library. We have human CRISPR Brunello knockout library. So plenty of products that we can support you with. But when we talk about the complete knockout, what is it? What we are talking about essentially is that we are talking about the double-stranded break. So it's actually that will allow to actually that will create actually a break within a double-stranded break within the sequence that will lead to a homology-dependent repair or a non-homology and jointing. And for the last one, so this will actually include or induce an error-free repair or an error-prone repair. For the first one, the homology-dependent repair, so we are looking at repair of donor plasmid or repair of sister chromatid. And I mean, when we talk about error-prone repair and repair of donor plasmid, so this is targeted mutagenesis. So this is the complete knockout. This is an illustration actually of, it is, oh, where did it go? Yeah. Okay, yeah, this is how does it look like, the PAM, of course, sequence. So this is, as you know, so this is needed actually by, I mean, it's the sequence that is recognized actually by the endonuclease during the editing. So if we are looking, or you are looking at increasing the expression, of course, so we can support you with plenty of products. But here I would like to highlight one specific product, which is the Mission TRCF or whole genome pooled and arrayed library. So here it will allow you to explore over 14,000 genes. It's a barcoded library. It performs CRISPR or RNAi rescue screens for full gene validation. It's ideal for difficult to transfect or primary cell lines. And here you can select your own promoters as well, so selection markers, fluorophores, and more. And this is so how to shows you how to increase actually expression. So here, as you can see, so see always, I mean, the PAM sequence, but the VP64 is the transcription factor that will allow actually to increase the expression of your gene of interest. If you are looking at decreasing your expression also, so we have several products that can support you with that. So TRC, lentiplex, pooled, shRNA libraries, CRISPR-I whole genome, and sub-pooled libraries, etc., etc. And here, so as you can see, so if you're looking at the whole genome, so we have the lentiplex pooled whole genome libraries. So if you are looking at a specific pathway, so we have the lenti-elite arrayed screens. If you are looking at gene families, pre-designed panels and sets, and so on and so forth, until we go down into individual clones. And to watch the time, so I do not go over time. So CRISPR-I, so for the inactivation, so and of course, so the CRAB, so which is a silencer actually that will allow the decrease of the expression. Sorry, decrease of expression of your gene of interest. So second step, evaluating and validating, of course. So it's the hit picking. So you will be selecting the. I mean, it goes through the hit picking and then validation. So you will select the number of guides, number of genes, the gene pathway of interest, the custom cloning. If you are looking at custom cloning individual targets from pools, and for the validation, so of course, so the delivery mechanism, so DNA, RNA, or RNP, a virus, and there are the assays that can be used for validation, like real-time PCR. Or real-time reverse transcription PCR, fluorescence, mass spec, or sorry, single cell analysis. So here, so for validating the CRISPR, just to give you a brief overview about what can be done. So you have the very basic stuff like the T7E1 digestion. That is actually it's a very cost-effective technique, and it will always be used as a first step before moving into another step like Sanger sequencing or next generation sequencing that can take from 2 to 14 days for Sanger sequencing to give you a result, and 3 to 5 days for the next generation sequencing. But of course, as you know, next generation sequencing is still not as much affordable as possible. So for now, but Sanger sequencing will always be the something that is affordable. I mean, the most affordable in this case. So creating your model, here I know that all of you are zebrafish enthusiasts, but of course, I mean, you can also pick your cell lines. So you choose your guides in Cas9 and then transfect and validate the RNP activity. And then so you confirm the targeted mutation. And then you can, of course, here, so go to, work with your, move into your model. So either this 3D cell culture or the 2.5D cell culture, and then to your experiment. So this is basically about the screening and then assessing your model. So it's always about the evaluation and the creation. So it's a circle of evaluation and creation, and see actually what is the impact of the technology that you have chosen and what you have, of course, I mean the targets that you have chosen as well. So this will help you to assess. It's like I said in the beginning, it's a circle that goes from design, evaluation, creation, and assessment. So, but after that, of course, when you have a model that you are happy with, so of course you will think of moving beyond the bench. So translating. So it's basically so scaling up using in vivo models, demonstrate safety after, of course, validating all of the previous steps. And for this, so so you have, I mean, if you move into the preclinical studies and the phase 1 of your clinical research or clinical studies, so you will need actually something that is more than a research grade. And here, so I'm just mentioning the pure edit reagents, which have been tested actually for their performance, for their safety as well, and to be used, as you see, so in the phase 1 stage of the clinical research. So, of course, I mean, here we are talking about the cell therapy discovery, because this is why, like I said, so one of the things that we want to do is develop therapies actually for the diseases, the existing diseases. And here, so of course, so you go from target discovery to cell engineering, cell characterization, development of specific media and reagents, the testing services, the need of the viral and gene therapy manufacturing platforms, and single-use assembly. So it's lots of things to keep in mind if you are moving into the translation phase, of course, and scaling up your, hopefully, I mean, the therapy that you are looking at developing. So, very briefly, so an overview of the workflow: identify and validate the target. And yes, so like you have seen, so you can see so pooled and arrayed screens, PCR, real-time PCR, nucleic acid purification, antibodies, cell assays, next generation sequencing. Create your model so we can support you. Here, of course, like I said, so you're looking at zebrafish, but also you can, if you are planning on using 3D cell culture or 2.5D cell culture, so we can support you, and even organoids in this case, so we can support you with that. Evaluating your model, so there are plenty of things that you can do, from actually the very basic stuff like Western blot into the more advanced technology like the chip arrays or mass spec. So, and we can also use immunogenicity assays, toxicity assays, etc., etc. And then, so of course, I mean, the translation, and so it goes beyond actually the step of the research and development, which would need vector scale-up, for example, mRNA production, bioproduction, etc. So this is the last of the end of my presentation. But here, so of course, so we have actually plenty of services that we can support you with: bioinformatics support, custom lentivirus production, custom pools, next generation sequencing, target ID and validation. So this is we can help you with all of that. We have I know that there are lots of attendees not from Tunisia only, but from overseas as well, which I would like also to welcome to the seminar. So let me know if you need any support. So I'm here, I'm based in Tunisia, take care of Africa and the Middle East, and I have lots of colleagues who will be all over the world who will be happy to support you as well. So, and that was it from my side. Thank you very much for your time.
Thank you very much, Dr. Midad. It was a nice presentation indeed, and very informative. So, any question from the audience? I have here a question from Badia Asghaya. If she's trying to understand or try to ask, how can we validate the safety of CRISPR approach for clinical purpose?
Yes, it's my question.
Sorry, how can we validate? Sorry, let me stop sharing.
The safety after the modification, after CRISPR approach, after we modify the genome, how we can validate or test the safety of this genome after the artificial modification by using the model or
Yes. So like I said, I mean, it needs, I mean, for any clinical trials, so you have to move, I mean, byte by byte from, like I said, from the normal, I mean, the normal models into the in vivo, so moving into animals, which is, I mean, for now also I know that there will not be a need to use animals, but still, I mean, you can always look at organoids, for example. So this is, I mean, it's critical before moving into the clinical, of course, the real clinical trials. But there are lots of things that can be used actually, or technologies to assess, for example, of course, the safety and the performance of the modified protein, like I don't know, like using, for example, assessing the concentration of specific biomarkers responsible for specific things, like, for example, for inflammation or things like that. So, I mean, before moving into the clinical trials on humans, which is something that we are still, I mean, not there. So we are not still at that stage. But before that, so before even moving to there, so there you can use organoids, you can use, plenty of, animals that, while we still can use them, so to test for efficiency or toxicity.
Thank you, Edith, and thank you for your nice presentation.
Thank you.
Okay, we have from both Vincenzo and Tom. So let's start with Vincenzo. Vincenzo, why?
Hello, thanks for the nice presentation, Wida. I just wanted to know, have you also available base editors and prime editors, which are like, the new generation tools which have been developed lately?
So far, we don't have them. We are working on developing few things that definitely will let you know about when they will be released. But for now, I don't have the information about that.
Okay, thanks a lot.
You're welcome, Vincenzo. I mean, this is Vincenzo. This is not going to be our last meeting, so we will. I'll definitely let you know. Hi, Tom. Nice to see you.
Hi, Edith. Thanks a lot for your nice presentation. I was curious. You talked a lot about genome-wide screening with shRNAs or guide RNAs. So, do you have specific tools for the zebrafish genome, or this is mainly for human genome screens?
Do we have specific things for?
For the zebrafish genome. So is this
for the
human
zebrafish?
cell lines or?
No, unfortunately, it's not specific to zebrafish. No, unfortunately, for now. I mean, I see that zebrafish is becoming more and more used. So maybe we'll develop also something specific for zebrafish. But yeah, unfortunately, for now, no, it's not specific for zebrafish.
But there's the option. I assume that you can have a custom library that you help designing, right? So if I'm interested in a certain pathway in zebrafish, I could get in contact with you and set up.
Yeah. Yes. So thank you very much for mentioning that. So thanks to the custom actually service, so we can support actually with the custom design for specific models. So this is something that we'll have to go through the custom actually process.
Thank you very much.
Welcome.
Any other question from the audience, maybe? Some rare genetic disease. Mariam Ben Amr, so can we use CRISPR-Cas technology to treat some rare genetic disease? So as I presented earlier. So yeah, so gene editing, zebrafish, and other small models are very, useful to understand rare genetic disease and to find solutions. So yes, it's a very, very helpful tools. And I do assume that in the next coming future months and years, there will be a lot of improvement in technology that could help patients. So yes.